So today's talk is, again, a revolutionary idea. They come from anywhere, but to research them properly takes time and money. And today we'd like to introduce you to one of the organizations that helps fund science and their new ideas for people earlier in their career. You can see the banner here from the Banting Research Foundation, a group not quite as old as RCI, but almost. And I'm sure you all know a little bit about the founders of the Banting Research. Presenting the Banting Research Foundation and introducing our three early stage researchers today is Dr. John Flores. John is a professor of medicine at the University of Toronto and Canada Research Chair in Integrative Cardiovascular Biology, researching circulatory and cardiac control mechanisms in health and disease, with particular emphasis on heart failure, hypertension, kidney failure, and sleep-related breathing disorders in men and women. John is a graduate of the UT Medical Medicine and a Rhodes Scholar at the University of Oxford. He completed a Royal College Fellowship in Cardiology at the University of Toronto, and then did postdoctoral research at the University of Iowa, eventually bringing his considerable expertise back to Toronto General Hospital and now Mount Sinai Hospital. Dr. Flores served for several years on the board of the Canadian Hypertension Society, including its president. His present administrative responsibilities include Deputy Physician-in-Chief for Research at Mount Sinai Hospital, Department of Medicine, and Director of Research at the University Health Network and Mount Sinai Hospital Division of Cardiology. Between 2009 and 2015, he served as chair of the Board of Trustees of the Banting Research Foundation, Canada's oldest medical granting agency, of which you will learn more this afternoon. John, I'd like to have you come up and say a few words. Thank you.
Thank you, Peter. We really are grateful to have this opportunity to present jointly and to bring together our two organizations. The Banting Research Foundation is a registered charity that funds health and biomedical research in Canada. We welcome this opportunity, as indeed for the last 90 years there have been quite a number of links between our two organizations. We were created by an initiative by Sir Robert Falconer and Sir William Mulock, who were at the time the President and Chancellor of the University of Toronto, and other prominent Royal Canadian Institute for Science supporters. And the current chair of our independent grant review panel, Dr. Reinhard Reithmeier, is in fact now the vice president of the Royal Canadian Institute for Science. We were established in 1925 to support the research of Sir Frederick Banting and other scientists across Canada with the hope of alleviating human suffering by inspiring scientific discoveries equal to that of insulin. The Banting Research Foundation now focuses on funding early career researchers who had the same challenges of Banting, who have innovative ideas, and we fund projects in all areas of health and biomedical research at universities and research institutes in Canada. In Canada, the transition from postdoctoral fellow to independent investigator is no easier now than it was in Banting's era. However, the resources required to initiate new lines of investigation are often orders of magnitude greater than they were at that time. Our mission is to invest in the early careers of researchers who demonstrate excellence and creativity in health and biomedical sciences. The Banting Research Foundation provides peer-reviewed Discovery Award grants to assist independent faculty initiating their careers at a university or research institute in Canada. These individuals have modest resources, but a passion to pursue imaginative, exciting, yet well-grounded new lines of medical investigation. Since its inception, we have awarded more than 1,300 such grants to support a broad range of health and biomedical research projects across Canada. And some of these awardees have gone on to have spectacular careers and create important new discovery. With national peer review granting agencies constrained in their capacity to provide such early career support, an increasing number of young investigators turn to the Banting Research Foundation for seed funding. Last year we received 50 such applications, but we had funds only able to support 6. This afternoon I have the pleasure of introducing 3 of the successful grant awardees. Our first speaker, Michael Suits, is an assistant professor of the Department of Chemistry and Biochemistry at Wilfrid Laurier. He will speak to protein structure-function relationships of periodontal disease. Our second speaker, Caitlin McLaughlin, is Assistant Professor of the Department of Psychology at the University of Guelph. Her topic is the evaluation of novel neurobiomarkers in the identification of adults with fetal alcohol spectrum disorder using portable eye tracking and EEG technology. And our final speaker, Noel Miller, is also an assistant professor at the Department of Psychology at Wilfrid Laurier University, and he will be exploring neural mechanisms of social behavior using zebrafish. So 3 very interesting topics, and I hope that they will be of great interest to those of you in the audience who were challenged by the weather and yet succeeded to come out today. Michael, our first speaker, received his PhD in biochemistry at Queen's in 2007, working on the structures and mechanisms of heme recognition and degradation proteins from a strain of E. coli. Before joining Laurier, he was a European Molecular Biology Organization Fellow at the York Structural Biology Laboratory, and then after that at the University of Victoria, where he was a Michael Smith Foundation for Health Research Fellow. Michael, we look forward to your presentation.
Thank you. Thank you. Whoop. Thank you very much for this invitation to talk with you about science today. So thank you, Kirsten, Ramona, the Banting Research Foundation, and the RCIS for, I guess, supporting my research. And I'll kind of discuss a little bit more towards the end what it's meant. So I'm talking about some of the structure-function relationships of proteins that are specific for bacteria that are growing in your mouth. And I decided, since it's so snowy out there, and I thought to reward you a little bit for coming in, that I'd go and I'd bake you some cookies. So I then thought, you know what, this group's gonna know something about science, so I better research some of these ingredients. So where do you go when you want, well, peer-reviewed material? Well, you go to Facebook. So I bought flour. Flour is not very good, I guess, because it contains a protein called gluten. So I thought, I can't put that in my cookies. And then I thought, what else? We've got eggs, and we've got butter, and those contain cholesterol, and I don't want you succumbing from cardiovascular disease, so that's also off the table. Then I got, you know, towards the ingredient end, we got sugar, and that's one of the big villains right now in our diets is sugar. So that's off the table. And the last was chocolate chips. And actually, no one had anything bad to say about chocolate on the internet. So I figure if you want, you can come up after and I can share some chocolate chips with you. But I didn't get through to bake these cookies, unfortunately. What I'd like to return to is this notion of carbohydrates as being bad. And I guess I'll use this kind of discussion period to, I think, educate you a little bit about carbohydrates, and not just sugar, but carbohydrates in general. And they really have this special place in our lives. They're the sweeteners in our coffees and teas and additives in cookies that make them even more delicious. They're in breads and cereals, things that we'll eat on a daily basis in order to round out a lot of the nutrient requirements that we need to keep active and keep healthy. And then as well, they're that kind of savory, sweet thing that you covet that you maybe sneak away and eat at lunchtime, or go a little too overboard with and end up putting on weight around Christmas time. But the idea is carbohydrates excite our brain. And that's an important thing because it's a resource that we covet, not just because it's sweet, but the reason is we love it because it's sweet. And the nature of that sweetness are carbohydrates. Carbohydrates are one of the 5 classes of macromolecular molecules that are important for life. And you can consider them as building blocks. So what I've put up here is glucose, which sucrose is it's composed of glucose and fructose. And glucose would be then a building block, a molecule that is part of that sucrose molecule. And there are 6 carbons on it. It's very similar to a kind of Lego building block, in that we can stack them together in various ways. You can make various linkages between a glucose molecule or other carbohydrates in order to make very complex structures. And if we look at then a bunch of hexasaccharides, carbohydrates that have 6 sugars, they're subtly different. So here's glucose on the left, and then what's called alpha D mannose in the middle. And you can see there's really only a small difference at this one carbon position with respect to a hydroxyl group. But yet that makes a big difference between your body recognizing glucose or recognizing mannose. And similarly, even in a slight distortion around what's called the anomeric carbon, whether it's axial or equatorial, makes the difference between it's an alpha or a beta mannose. And you can see how we could get a large complex of those kind of building blocks stacking in various ways just by even looking at these three, what are called monosaccharides, single carbohydrate molecules. But if we consider them as building blocks that we can stack together in various ways, we can start to make these really amazing structures, just like you would with Lego. So here is an amino acid, asparagine. And when you link two carbohydrates to it, you can start to build up what are called N-linked glycans. And so these coloration will kind of depict different carbohydrates as well as the coloration themselves. This is an important decoration on many yeast proteins. It's called mannan. And really, if we looked at it back at that Lego building block level, you could take carbohydrates and basically make a structure like you would see in your children's room or your grandchildren's room. But what's important is it gives us a really important barcode. So if we just return to some of the complex structures here, it's even kind of complex for our eyes to see. But this three-dimensional information, as well as the electronegative fingerprints that are present on those carbohydrates, make this a really important thing, that it really will give your body and give bacteria and viruses that are interacting with proteins a real signature that they can identify. And it's really typified by blood group antigens. So here I've got up a kind of cartoon depiction of a red blood cell, and it will be decorated with various groups. So if we look at the O group here, these are carbohydrates that actually confer that O blood type. If we look at the A group, it's very subtly different. It's got one carbohydrate different, and just like the B group, it's now a slightly different carbohydrate, and yet we all understand that if you were to receive a blood transfusion of the wrong type, it would be rejected, and you would have large immune response against that. And it's really this subtle difference at a carbohydrate being there or not, or being a universal donor. And so you can really see how carbohydrates form the basis for a lot of important interactions, not only at, say, the blood group level, but as well as an important signature for when things are recognized. And so if we look on the cell surface, be it your cell or an invading virus, bacterial cell, a lot of the interactions that are mediated between, say, antibodies and surface recognition, protein-protein interactions, or, say, viruses and bacteria interacting with your cells, they're actually mediated by carbohydrates. So for good or bad, we're kind of stuck with these things. We need them, and that's part of why we get excited when we taste something sweet, is because it's providing us with these building blocks that we'll use in order to build complex structures that allow cells to communicate. They won't see, but they can at least reach out and kind of share information via those carbohydrates. And things can get really complicated. Here's the major classes of carbohydrates, and some of them you may have heard of. There's those kind of structures, these N-linked glycans that I depicted a little bit earlier. They're often appended onto proteins in the form of proteoglycans, or they're long linear chains, things like chondroitin sulfate, hyaluronan, heparin sulfate. They're long linear carbohydrates that confer important cell interactions. And so as a kind of biochemist focused on carbohydrates, I started to kind of think of important places where carbohydrates are operating. And if you ate that cookie, what would happen next? You'd take it into your mouth and you'd chew it up and it would, you'd swallow it. But there would be some residual material that would be left in your mouth. And I thought, I guess when I was doing my postdoc, there was a lot of research going on in the gut, and we started to appreciate a lot of the things that were going on between bacteria interacting with the multitude of bacteria present in your gut. And I thought a new avenue may be to look in the mouth, which it may seem almost too obvious to look there, but it seemed an interesting idea. And with the Banting Research Foundation supporting some of this research, I started to delve into dental research and dental health. And despite everyone kind of dislikes going to the dentist. It's an unpleasant experience, but it's actually a little bit more of a serious disease. If we look at the amount of people globally that are affected by dental disease, it's about 1 in 3. As well in Canada, and this is not just treatment of disease, but also prevention, so it's a little inflated, but we're spending a lot of money on making sure that our mouths are in good shape. I mean, our teeth are important, and looking after them is actually a little bit more important than we thought, and I'll show that on the next slide. So this is a nice, healthy tooth. It's supported by ligaments. It's well innervated, and it's protected by the gum. You're going to have a certain degree of bacterial degradation via acid hydrolysis of the actual tooth itself, but then you've got these nice remineralizing osteoclast, osteoblast cell that will actually give that bone more structure. So this is the nice, healthy example. And what's interesting is there's a state called dysbiosis. So I guess homeostasis would be where everything's operating nicely and your bacteria and other things are interacting with your own cells in a kind of proportional way. In the case of a dysbiotic system, you've kind of gone to a place where that gum is now receding. You're going to have not as well supporting tissues around the tooth itself. And as well, it can result in demineralization, cavities, dental caries. So what's kind of interesting is this is happening all the time, and yet in certain individuals, the scales will have tipped towards this dysbiotic state. And it could even be on a single tooth or a single region of your mouth, but it's tough to turn back the clock and regain that healthy state. And the nature of my research is to understand kind of the ebb and flow of what would be the wrong set of circumstances to result from a healthy individual with a well-supported tooth to that dysbiotic state. And what's interesting is the mouth is a bit of a canary in the coal mine in that if you're having dental disease, it's co-indicated with a large number of other severe disorders, things like diabetes, cardiovascular disease, adverse pregnancy outcomes. Migraines actually were linked about a month ago in a publication. And I think it acts like a canary in the coal mine in that it's a training ground for your body to interact with bacteria in many respects. And if you're not able to interact and play nicely, both the host and kind of the microflora that's existing in our mouths, if that's not operating and it's in that dysbiotic state, it can be endemic of a more systemic problem. And so what's interesting is you have about 700 species of bacteria that grow in your mouth, and a lot of them, they're not really good, they're not really bad, they just kind of hang out there. But around 98, there was a study that actually said, what's interesting is there are bacterial co-conspirators in that when we look in dental pockets and various places within the mouth, there's bacteria that seem to associate together. And so I've depicted some of the classes that Socransky identified. And the big one that I'm interested in is this, what's called red complex. 3 bacteria, Porphyromonas gingivitis, Tenorella forsythia, and Treponema denticola. And these, when they're associated together, there is a larger incidence of dental disease and dysbiosis. So we're going to look at the way that these bacteria interact, how they communicate, and it's almost akin to if you were to, say, want to rob a bank. Not that you would, but if you were to, say, write a movie where you were going to rob a bank, you would need the driver, the person that can kind of get you to and from the place. You need the person that's going to be the heavy, that's going to maybe scare the people into, I don't know, lying down on the floor. And then you need the brains, the safe cracker. Any of those in itself, you're not going to successfully rob the bank or, I don't know, entrench and cause dental disease. But together, when you share those resources, those abilities, that's where you're going to be able to maybe successfully pull off the heist. So it's very similar to this red complex that each of them will bring different traits and different properties, and it's really the strength of the 3 of them that would cause that dysbiosis in the first place. And so as you look at the surface of a tooth, it would be similar to building up a little environment for bacteria to live in. You're going to have maybe not just the red complex there, but some initial colonizers that'll build up what's called a biofilm, the plaque, the stuff that your dental assistant really needs to scrub to get off your teeth in order to get rid of that. And if we appreciate that proteins are responsible for recognizing some of the composition of those biofilms, and as well will build up those structures in itself, we're hoping to understand a little bit better about how that operates. And I think I'm short on time, so I'll just say that what we do is we'll look at individual enzymes, and we'll look at what's going on in the active site where they'll recognize carbohydrates and break them down, or at the corollary end where they're building up biofilms, the synthetic machinery that'll be synthesizing those. And in a way, by studying kind of individual components and by starting to build up our understanding of how enzymes act on individual carbohydrates and some of the specificities of that, we could start to understand how a complex structure like an N-link glycan could be degraded by bacteria. In this case, it was strep pneumonia, something that I was studying before. But as we built up our understanding of enzymes that are present in that red complex and some of the factors that are produced, we're hoping to maybe understand the disease and maybe prevent it. And this was a kind of gross example of dental plaque that's present on teeth. I don't know how long this person hadn't brushed their teeth for, but the notion is you could maybe get from a state, hopefully not that bad, but a state where you're in that dysbiotic, over kind of immune response, and with the right chemicals and conditions, maybe targeting specific enzymes, or maybe just taking things that are common in mouth rinses, you may be able to go back to that naive state. So whereas someone, in this case, despite kind of maintaining good oral hygiene and brushing their teeth, flossing, regular professional care, if you're in a state where you can't actually remediate that because of that entrenched bacterial, maybe red complex, if we find the right set of conditions, maybe we could gain back some of that naive, healthy dental state. And so with that, I didn't really talk about any of my research in particular, but we're building up our understanding of what's going on at the bacterial level. And in part, the Banting Foundation was really insightful and really timely in that it took my research in a new direction. And with an initial seed grant, which we then parlayed into a second grant and now a third grant, we're now over $900,000 in support. And in part with the Banting Foundation kind of recognizing us and also us recognizing that it was kind of interesting for us to be studying in the first place, it wouldn't have been possible. So I'd also like to thank Laurier as well as Glycocode, a commercial partner Mrexis, CFI, and NSERC for foundational support. So thank you very much.
Thank you, Michael. Michael and the other speakers will be up at the end for a question and answer period. I'd like to move on now to our next speaker, Caitlin McLaughlin. Caitlin is an assistant clinical professor in the Department of Psychiatry and Behavioral Neurosciences, an associate member of the Department of Psychology, Neuroscience, and Behavior Department at Guelph and McMaster, respectively. She completed her MA and PhD in clinical psychology with a forensic specialization at Simon Fraser University, and then was subsequently awarded a training fellowship by NeuroDevNet and completed 2-year research postdoctoral fellowship with concurrent appointments in the Departments of Pediatrics at the University of Alberta and the Child and Family Research Institute of Developmental Neurosciences and Child Health at the University of British Columbia. She'll speak to her interesting work on fetal alcohol syndrome disorder. Caitlin?
Thank you. All set. Wonderful. Thank you again, everyone, for coming for this afternoon, for the invitation here to speak. I think knowledge translation is a really exciting part of our career that we don't get to do as often as we would enjoy, and especially to the Banting Research Foundation for funding the research that I'm going to tell you about today. I was going to contribute a bottle of wine, given the theme to our topic, but it's not quite 5 o'clock, and here anyway, and the REBs really do give us a hard time around doing research with alcohol in this population. By way of background, I'm a clinical forensic psychologist, so my research really is at the intersection of anywhere that clinical disorder, mental health issues, or substance use, in this case fetal alcohol spectrum disorder, intersects with the criminal justice system. So for those who aren't familiar, FASD is an umbrella term that really refers to a full spectrum of disability that occurs after a woman drinks alcohol during pregnancy. FASD comprises a range of adverse developmental outcomes with a quite heterogeneity in clinical presentation. Broadly, people experience significant problems in neurocognitive impairment, so neuropsychological problems, problems in their emotion regulation and behavior. And then in a much smaller proportion of cases, you'll see external physical features of the disability, where you see some facial dysmorphology changes, in particular to the shape of the face, that actually occur very, very early in a pregnancy, typically before a woman might even be aware that she's pregnant if she's not actively monitoring her cycles. As well as, there's a little bit of controversy over this at this point, but there's evidence to show that in the early stages developmentally, you see changes in growth stature, so smaller size. We were, as a panelists, trying to think about how to link our talks because they're on such different topics. And actually, the last talk we're going to hear today is on zebrafish. And zebrafish are one of the biological models that we do a lot of preclinical work on in trying to understand the impacts of prenatal alcohol exposure. So I wanted to show you just a picture of what happens when you put the little zebrafish embryos in a petri dish full of alcohol. You see major significant physical changes. But problematically, in about 90% of people affected by fetal alcohol spectrum disorder, there's no outward physical signature. It's a bit of a myth that you'd be able to see somebody with smaller eyes or a change in shape to their face. Very few people actually by adulthood are small in stature, and so we think of it as an invisible disability. People with the cognitive problems develop compensatory strategies, and they can kind of talk the talk. And alarmingly, people really make it a long way through the lifespan without being detected in clinical service. In addition to the circumstances that would often be associated with a woman continuing to drink alcohol or use other substances in pregnancy, these children are often raised in environments marked by significant additional environmental adversity. And so sometimes you really see a confluence of the most disadvantaged young people from prenatal development, right from the initiation of pregnancy all the way through the childhood and adult lifespan. So you see exceptionally high rates of trauma, of poverty, of educational disruption, of early life abuse. In foster care populations in Canada, they estimate that as many as 50% of children may be exposed to alcohol prenatally. So we really end up seeing exceptionally adverse developmental outcomes, poor outcomes for this population. Which brings me to my area of work. If you think about the prevalence of FASD in North America, I'd say pretty conservative estimates suggest that anywhere from between 1% to 5% of the population would present somewhere on the spectrum. The thought is that this is likely an underrepresentation and a pretty conservative estimate. This is a difficult disorder to study epidemiologically. Alternatively, in criminal justice settings, the few studies that have been done estimate prevalence rates ranging from between about 10 to 23%. So for instance, I went up to the Yukon to do a project with the government there over a few years, and we did a sampling and estimate of the population prevalence in corrections of FASD and found about 17.5% of offenders there met diagnostic criteria, and that as many as 30% could have. I'm going to talk a little bit about some of the clinical complications involved in making a diagnosis of FASD. But the one hallmark of the diagnostic process is that you have to be able to demonstrate that there was prenatal alcohol exposure. Particularly for adults, this can become increasingly difficult if a biological mother is not in the picture, if the relationship's not intact, if there's not a family member who can speak to the fact that the prenatal alcohol exposure occurred, then we can end up having significant neuropsychological or behavioral signs and not able to confirm a clinical diagnosis. And again, I think these costs are conservative, but one of the key drivers, I think, behind health science research these days is what is the cost to society, what's the economic impact. And we know that FASD accounts for an exceptionally large amount of healthcare costs, but you can see here that one of the biggest drivers at this point in terms of Canadian estimates is the cost of FASD in the criminal justice system. And that's because it's estimated in both the US and Canada that anywhere from between 60 and about 65% of people with FASD at some point will come into contact with the criminal justice system. So why is this relevant as a forensic and clinical researcher? FASD, though the research, the empirical body of literature, I think, is really just emerging in this area. It's linked with some really problematic, dangerous, and costly problems that we have to grapple with in the correctional environment. FASD is linked with high rates of self-harm and suicide, victimization, aggression and violence, and also recidivism. And frequently, people with FASD are thought to really become trapped in what we would call a revolving door problem in the criminal justice system, where they're arrested, they're charged with a defense, they breach conditions or are charged with another offense and subsequently end up becoming really institutionalized. As I alluded to earlier, screening and diagnosis for FASD can be very challenging. This is in part because around 90% of individuals would not necessarily present with an overt physical presentation that you'd be able to recognize as a clinician without a lot of education in the area. So sometimes they can be difficult to spot. There are, at this point, no clinically validated screening tools that would really work across settings in helping us pick up on people who are at risk of having the disability. The average assessment involves an interdisciplinary evaluation that's not dissimilar from autism and is thought to cost through private clinics between $5,000 and $7,000 per evaluation. And there are very few clinicians working in criminal justice settings with at-risk populations who are also experts or have the expertise to do screening and diagnostic work in fetal alcohol spectrum disorder. There's also a stigma that continues to be associated with the disability, the idea that only bad women would drink while they were pregnant, and that people who have FASD are bad people. And some of that remaining stigma and blaming and shaming can really deter people from wanting to engage in a meaningful way in the diagnostic process. And so broadly, a failure to recognize and catch these folks in the criminal justice system, we think contributes to this revolving door problem and a very expensive and costly high rates of recidivism for the population. So my research at this point is turning to new novel ways of potentially being able to identify people early, ideally translationally, in their involvement in the justice system. And this research parallels work that's being done in children with FASD. So I'll tell you a little bit about some of that work. But just by way of backdrop, in case you're unfamiliar, I'm going to use the word saccade. And really what we're talking about here is quick movements that your eyes make that are often automatic and outside of your control. I won't go into a lot more depth of that. But basically, when we're looking into the environment, our eyes do a lot of automatic processing that involves a complex relationship and structure within the brain. And so measuring the way that our eyes make those movements can provide a really powerful tool that lets us see inside the brain, looking at motor control, cognitive functioning, and different kinds of sensory processes. So a saccade is really when your eye is looking toward the target that it means to be looking toward, so it's consistent. So if you look up here, you see there's a target here, I flash a target, it goes away, and I give you the instruction, look to where the next light flashes, and your eyes will automatically look to the next point. That's consistent with the instruction that I've given. An anti-saccade is the opposite. You'll be asked to look at a point, there'll be a gap, and then the instruction is, we're gonna flash another point on the screen, and instead of going in the direction that you would want to automatically go in, you're going to inhibit that response and look in an alternative direction. Then there's a third kind of task that we would think of as a measure of working memory. It's called memory sequencing, where we say we're going to flash an initial target, and then there'll be a blank or a pause. We're going to flash one, two, or three more targets. They're going to go away, and then when we ask you to move your eyes, you're going to follow that sequence around the board. And what we do is we use infrared cameras to actually measure a whole lot of metrics around the way your eyes perform these commands. And the metrics that we get map nicely onto measures of neuropsychological functioning that we'd administer in a clinical setting, for instance. So, in a number of studies that have been done, children with FASD show not only slower response times across trials, but they make a number of errors. And I'll show you some examples of those. So here was one study conducted by one of my collaborators some time back now. You can see here the healthy controls. Here's the target, the initial point we asked them to look at. And then here's the onset of the new target. This is an anti-saccade, so we've asked them to go in the other direction. So you see the controls, they kind of, oh, okay, let's move our eye. An ADHD comparative sample has a little bit of differentiation compared to the pattern in that group and then gets going. And then you can see in the FASD group a much longer response latency across the group and a lot more variability or error in terms of their accuracy in being able to perform the task. Importantly, eye movement control tasks have been correlated, as I mentioned, with lots of neuropsychological measures. So they correlate highly with deficits on tasks of executive functioning, including things like inhibition, set shifting, working memory and impulse control. They've been correlated with white matter tract integrity differences in the brain using DTI, and very recently altered neurological recruitment patterns using EEG. This is important because these monitoring paradigms I'm going to show you a picture are non-invasive and portable. And so if they correlate highly with more expensive and complicated tasks that require neuropsychologists, for instance, to administer, then being able to use them as an initial screening point might flag who needs to be referred for more in-depth clinical evaluation. One of the limitations so far in the literature, though, has been that often we're comparing the brain function of people with FASD to these supernormals. And so I'm going to tell you why that's important in the correctional population. I'm going to show you one more study and then tell you a little bit specifically about what we're doing for this. But this is a study that specifically paired portable EEG monitoring with portable eye movement control technology to try and differentiate children with FASD from typically functioning controls. So they did a memory-guided task. This is, for instance, they were asked to look at a point, and they were shown more points, and they had 1, 2, and 3 stimulus conditions. So they increased the difficulty across trials. You had to remember to look to more places. And they used one of the very first single dry channel portable EEG monitoring headsets to try and take a look at brain activation patterns while people were performing this task. And what they found was that children with FASD had fewer overall correct trials. So here's the correct trials figure, where the blues are the controls and the reds are the kids with ADHD. And you can see as the number of stimuli increase, they start to have fewer correct trials overall compared to the other group. And they also had more sequencing errors, so their eyes went in the wrong order or to the wrong target fixation point compared to the controls, particularly when they hit 3 stimuli. So that makes sense. As you make the task more difficult, the brain functioning is often thought to be less efficient in FASD, and kids start to have more difficulty, make more mistakes. Importantly, what they also found was different underlying patterns of neural recruitment, and specifically they showed reductions in alpha and theta power at increased load, so with more stimuli. So this is a figure specifically from looking at the absolute alpha power, and so they showed, you can see the differences in the purple lines between the 2 and 3 stimuli in the FASD group. This is the reference point, so right when they were showing the target point, and then these are the memory delay averages, and a significant difference between groups there. So what we are aiming to do here, and we're getting going very shortly, is really tackle this urgent need to identify neurobiomarkers and potentially cost-effective and quick screening applications in criminal justice settings. You can imagine that being able to bring an MRI or a $7,000 a pop neuropsychologist into a prison is quite difficult, and these are systems that are exceptionally strapped, both in terms of mental and general health care. So there's a need to create something that can kind of come out of the box and achieve this screening need to identify where to target the most the resources most effectively. So we're going to be using the iLink 1000, and that's a portable camera and screen that can be clipped onto an arm. We're going to fly it around all over the place. And then also a new patented Virtus Smart headset that actually has a 10-channel dry electrode system. So if anybody's familiar with EEG, you see, remember those old caps and the gels and everything gets stuck and it's very messy. So this is a 10-channel system that can actually be worn dry. It fits just like a headset, and you can wear it over a 24-hour period. It's actually been developed to try and monitor seizure activity at home versus in the hospital and reduce healthcare costs. We're going to be recruiting people with FASD in the criminal justice system, people in the criminal justice system who do not have FASD as a more ecologically valid control group, because we know that there are very high rates of cognitive problems and adversity in the criminal justice system broadly, but that people with FASD just present with them at much higher levels, and then also kind of a group of community supernormals to give a more valid comparator. And we're doing this work in collaboration with Dr. James Reynolds and Dr. Michael Amlung and Gary Shamowitz at McMaster University in Queens. And really what we want to understand are 3 things that we'll look at. We're trying to validate and replicate the use of these technologies, moving from the pediatric realm up to the adult world. We want to identify potential neurobiomarkers by combining these different behavioral eye tracking and EEG measures to see if we can come up with markers that could help develop screening cut points to help institutions actually implement these kinds of tools. And then also in this study to look at the sensitivity and specificity of doing so. And really, the future programmatic focus is to actually be able to do this at a much larger scale. So we're very, very thankful to have the money to do this pilot work, to be able to establish proof of principle in a complicated applied setting and also in adults. And then to be able to add a free viewing condition. New research is showing that if you show a video of about 15 minutes that has a range of stimuli that change in characteristic from bright to fast to dark, different color contrasts, the eyes, human eyes tend to move in a systematic and pattern-based way, and people with neurodevelopmental disorders, other cognitive problems, don't look at things in the same way. So if you look at a video for 15 minutes and record eye movements and get very high numbers of people in your sample groups, just by having people watch a screen, you may be able to distinguish who's at risk of having neurocognitive deficits from who isn't. So they're actually doing this in infants right now, and we think this may be an even simpler type of method to be able to do this with big kind of data mining and computational methods, to be able to add multiple ways to look at this screening technology. So that's where I'll leave you, and I just again want to thank very much the opportunity to speak with you today, and also for the seed funding from the Banting Research Foundation. Thanks.
Thank you, Caitlin. As a third example of the diversity of research that the Banting Research Foundation supports, I'd like to introduce Noam Miller from the world of collective cognition using the zebrafish model. Noam received a Bachelor of Science in Biology from Tel Aviv University. This was then followed by a Bachelor of Music in Performance from the Royal Conservatory of Music of Toronto. He then went on to receive an MA and PhD in experimental psychology from the University of Toronto, and then was a postdoctoral fellow in the Ecology and Evolutionary Biology Department at Princeton University before assuming his present position at Guelph. No? Wilford Lawyer, sorry.
Okay, is this working? Am I talking? Okay. Thank you very much for coming out, and thank you very much to the Banting Research Foundation for this grant that's enabled us to do some of this work, and to the RCIS for inviting me to give this talk. This is going to be a little different. There won't be very many humans in this talk. I mostly want to tell you about some research that we've been doing on how groups of fish behave, and I'll be focusing in on just one experiment that we've done that answers one specific question, but I want to emphasize throughout this talk that what the principles that I'm going to be talking about apply to all groups. The kinds of problems that groups of animals have to solve are often very similar, and these things apply equally well to groups of fish or groups of humans. And hopefully, I'll be able to make that relevance clear. There are a lot of different species that live or spend a lot of their time in groups, all the way from the eusocial insects through to fish and birds and mammals, including ourselves. And there are a lot of really good reasons to live in a group. There are a lot of advantages that you get from being in a large group. For example, large groups are better at detecting bad things that might be arriving, to the extent that in some species we have dedicated guards or bouncers. In our species, when you're in a group, it's possible to acquire information about potential mates by observing them dating other people. There are advantages to hunting in a large group, which allow you to bring down prey that a single individual would not be able to take advantage of, like all the dioramas of Stone Age people hunting mammoths. And conversely, when you're in a large group, you can sometimes defend yourself better against large and dangerous predators that might attack you. In some cases, you can actually directly share information between members of groups. You can tell your friends, for example, how exciting the RCI science talks are, and then they're more likely to attend those in the future. And we know that bees, for example, do this. They tell each other where to find food. And there are other various advantages, like, for example, moving in a group can sometimes be very advantageous, like bikers that use each other's slipstream to avoid having to fight the turbulence in the air, which is what these birds are doing here as well. And this is just a very partial list, and there are plenty of much more extensive lists of these advantages that have been published. And the way that we usually think about these various advantages is by dividing them up by what kind of advantage they give us. So some of these things help us avoid dangerous things in our environment, like predators. Some of these things improve our foraging or ability to find resources that we need. And there are several other different kinds of mechanisms that don't fall into either of those groups. However, one of the things that we can do is, instead of looking at what kind of advantage, or what we might call the adaptive function of these various effects, we can look at what kind of mechanism is going on in the animal's brain when they're doing these things. These kinds of effects over here are simply statistical effects of being a member of a large group. They don't require the animal to make any kind of decision short of remaining within the group, and they generally become more effective the larger that group is. On the other hand, these effects over here are situations in which the group merely provides information to the individual, and the individual can then choose whether to act on that information or not. And of course, there are things that don't fall into either of those categories. And so the question that we want to ask is, when animals make decisions about groups, which of these two things is driving those decisions? And the problem that we face is that under normal conditions, anytime an individual makes a decision to join a group, those two mechanisms are perfectly confounded. Both the safety in numbers kind of mechanisms and informational mechanisms both suggest that you should stick with the group. You should stay with whatever the largest group is. Those are some of my students. And so the question that we can ask is, when an individual makes a decision to join a group or remain with a group, is it simply a generalized, I want to be with the group decision, or are they actually relying on the information that the group is hopefully providing to them? And so in order to ask this question, we did this with something that I call the all your friends are idiots experiment. And the idea here is that if you're a member of a group, and that group consistently makes really, really bad decisions, things that are obviously bad, then you as an individual are faced with a conflict. On the one hand, the informational type mechanisms are suggesting that you should do the opposite of whatever that group is doing because they're all idiots. Or, as my mother liked to say, if your friends were to jump off a cliff, would you also copy them in that behavior? And she was hoping that I would say no. On the other hand, you are still safer, as a rule, being part of that group than you are on your own. And all of us have our own litany of bad decisions that we've made simply because everyone else was doing whatever it is that we really should not have been doing. So the question that we wanted to ask is, can we set up a situation where these things are in conflict, and can we set up an experiment where a fish would learn that it has to ignore the group and move away from a group in order to have some sort of positive outcome, in this case, finding food. And here's how we actually did this experiment. This is our experimental aquarium. We place a group of fish, 5 fish, in one side of the aquarium, and down here in the bottom of the aquarium, we place our one test fish. And all of these walls here are transparent. We leave the test fish in here for a couple of minutes to observe its friends. On one of the sides, on these little feeding rings, we place a little piece of floating food. And after this individual has watched its friends swimming around for a little while, we lower an opaque barrier, a wall down there, to block off its view. And we then raise this dome and release the fish in here, and it has to swim around to find the food. And in the key experimental condition, the food is always on the opposite side to where the fish are. So in order to get the food, you have to move away from the group. In this case, these are the idiots, and this is the fish making the decision. And as I'll show you, we have some control conditions, and obviously, from trial to trial, where these individuals are changes in a random manner, so that the only way to find the food is by reference to where the group is. So let me show you what happens. There's a lot of groups in this experiment, so I'll walk through them one at a time. To begin with, we call our basic experimental group the avoid group. These are the fish that have to learn to avoid the group in order to find the food, and they can do it. These are the results of some probe tests, and they're not great at this task, but they are better than chance, and so they can learn to ignore the group and move away from the group to find the food. Just to control for this, we also ran a group that simply had to approach the group. So for this group, the food was always on the same side as the rest of the group, and these fish also can learn to do that, and they're equally good at it. They're no better than the avoid group. So it was a little confusing to us, and so we asked, what happens if they have to ignore the group altogether? Can we teach these fish that the food is always in the same place? Doesn't matter where the group is, just go to that one side. We call this the side group, and they also are able to learn to do this task. There's going to be a litany of that. And so one thing that potentially bothered us was we said, well, this is a rather artificial situation for a fish. Are they even attracted to the group at all in this strange situation that we've put them in? And so we ran a condition that I call the both condition, where there was food on both sides of the tank, and so they could join the group or avoid the group or do whatever they wanted. And what we find in that condition is that they really don't care about the group at all. They're just doing whatever they feel like. Well, this was concerning, and so we wondered if perhaps the issue is that this wall that we've put down that separates them from the group is making them not care about the group so much because they can't see them at all. And so we reran the entire experiment again, and this is something that's a little bit still ongoing, but this time we just removed that large opaque wall that blocked their view of the fish. So now, when the test fish, which I haven't drawn here, is making its choice, it can actually still see the group at the time that it's making its choice. And what we find when we do that condition is that when they have to move away from the group in order to find the food, they're no longer able to do that. The impulse to join the group is a little bit too strong, whereas when they're required to join the group, to approach the group, they can still do that. And when there's food in both sides, they tend to choose the side that is close to the group. So I want to emphasize that this is not a problem, again, that is just faced by fish. This is something faced by humans as well. This is from an old Japanese game show. Pay attention to this woman here. She is the unsuspecting subject of this prank. And she follows the decision of the group. And again, what we're trying to do here is tease apart why she is following the decision of the group. Is it simply because everybody else is doing something, she also feels that she has to do it? Or does she think that the group actually has some piece of information that is beneficial to her to act on? And what I hope I've shown you is that with fish at least, as with humans, it's a little bit of both that is going on. And so what we conclude from all of this is that yes, the fish do have a strong tendency to join the group. They prefer to be with the group for obvious statistical reasons, especially if they can see the group at the time that they are making decisions. Then the peer pressure to conform to the group is a little bit too strong for them to counteract, even if this means that they end up not getting fed in the experiment. On the other hand, if we hide the group at the time when they're making their decision, they're able to overcome this innate tendency and to be antisocial when that is actually beneficial for them. And anecdotally, I can tell you, just to bring this back to humans one more time, that human beings also make much better choices when they're not in the company of a large group at the time. It's easy to avoid mob mentality by simply removing yourself from the mob. And so this is the line of research that we hope to continue to develop to show these analogies between how fish solve these kinds of social collective problems and how human groups also function. I just want to end by thanking the many members of my lab. These experiments take forever to run, and all of these people were extremely helpful in doing that. I particularly want to single out Rami Ayoub, who's a student in my lab who did much of the work for this particular experiment. And of course, I want to thank the Banting Research Foundation and NSERC for funding my research. Thank you very much.
So we have the three speakers come up, and we'll have some questions. And also, if you're prepared to stick around for a little bit afterwards, I think there's coffee and.
I'm having chocolate.
I did promise, yeah.
So, questions, comments?
Is there any indication that vaccination would be a useful strategy against all diseases in your work?
I can't think of where I've read where vaccinations are used in a kind of prophylactic way to prevent dysbiosis, but I think it would probably work in certain examples. But when you're inoculating, typically with a vaccine, it would be against one organism. But frequently, the agents that are responsible for that dysbiotic state, they can be varied. So they're organisms that we understand very well. Strep mutans is a good example, and Porphyromonas gingivalis. But just inoculating those, like a good example is we probably have those agents in our mouth all the time. And just by maybe knocking those down or by preventing them from getting entrenched, we still don't understand. So part of my research is to try to tease out a deeper understanding of that.
Yeah, a question to Noam. Are there similar studies with other animal types, other animal, what do you call it, classes or kingdoms, or not kingdoms, or, like mammals, reptiles?
Right. I mean, there are studies on how you decide whether to join a group or not, or which group to join. Those have been done across a large number of species. These kinds of questions of putting the two mechanisms in conflict haven't been done very much, but there are quite a few studies on humans doing these kinds of things. The canonical thing that I think of this being is a little similar to is the marshmallow study, the kind of delayed gratification in children study where your impulse is to eat the marshmallow that's in front of you right now, but there are cues that are telling you should actually hold off and that'll be better for you later. And we know that, again, the younger children are not so good at inhibiting this, and there are differences between individuals. And one of the questions that we're addressing is, can we change this in different populations of the fish, potentially with drugs and other ways?
You all there was an appearance of brilliance in all of your diatribes. You didn't quite put a total muffler on the audience, your friends, in trying to strive for or make a reach out to formulating an axiom of excellence in regards to your research and your viability and your delivery of some of your speeches from your lectern. You talked about safety in numbers and people in a group joining the hierarchy, and then anti-social behavior and behavior that inspires in the ethical classroom as being a stimulus, and the whole the foliage of chemicals and the understanding of the scientific and psychological artists in you, and your innate qualities that you've developed in your students, and how you put your spotlight on your exceptional students and your research. I was just wondering, what axiom can we get out of all of this? That proposition that right now that is kind of hidden in the hidden curriculum and pedagogy of what we're talking about. that could affect the arts, musical talk, the sciences, philosophical breakthroughs that we're just dwelling on now. The ever-present axiom that we're coming out of the jet to the cave, and we're going to the paradigms around the corner. Can you not embellish it, but can you capsulize that axiom?
I'll take a stab at that. I was also struck, actually, by the absolute comedic brilliance and also the balance of seriousness and entertainment value, because it's such a difficult skill as new researchers to be able to figure out how to become a scientist and figure out how to become a teacher and figure out how to get grant funding, and then also to be able to somehow communicate all of this with clarity and accessibility to an audience. So I really enjoyed that piece, and I think there was some artistry here with my colleagues. I also was thinking about the cross-application between our different, completely, at first sight, completely different bodies of research. But what was interesting to me is that in FASD in particular, we have exceptional changes hypothesized to the synthesis and composition of gut microbiome communication, higher rates of disease across health areas, including, I bet, periodontal disease, as a result not only of poor prevention, but all kinds of changes in the system. And we're actually doing research right now looking at delay of gratification and social group decision-making in FASD, because that's one of the areas of executive functioning and social cognitive interaction that seems to be exceptionally impaired in our population. And so you can see that with basic research and clinical research, I think there are lots of shared mechanisms. And for me, to I think maybe speak to your point is kind of what's the theme, what's the point, is that by working together from sometimes completely different disciplines, we can come up with completely new mechanisms and ways to investigate important preclinical and clinical questions. And so that's what struck me, if that helps you. That's what I was thinking about. Anybody else?
Just want to pick up on that and maybe have you just, the three of you, sort of talk about your challenges as early stage researchers. I mean, what has been some of your bigger challenges as you try to do teaching and science and funding?
I think those are important resources, but I think you can see you need to wear many different hats starting out in research, and the teaching and grant funding, and as well being a colleague. And so it's really time is, I think, that the final limiting resource. And so managing that well and finding mechanisms to make yourself get over the humps when you're not feeling up to doing something. But I think everyone's path is a little bit different. So no one can give you like a nice blueprint. You kind of need to get comfortable in your own skin and get over a little bit the imposter syndrome that you don't belong. And then once you get over those things, manage your time, and that's where
I would add to that to actually say something positive about our funding agencies, which hopefully will shock you all. That I think one of the challenges, at least that I faced, is having complete freedom over your research agenda, not being in a lab where that's being set by somebody else, or at least guided by somebody else, and coming up with a research program that is, as I think my colleagues have done really wonderfully, coming up with a research agenda that is broad enough so that there's a lot of work to do and questions to answer, but narrow enough that you don't spread yourself over too large an area. And one of the ways I think that funding agencies help us with that, perhaps unintentionally, is by forcing us to write these rather detailed proposals where we say, okay, here's what I can realistically achieve within the lifetime of this grant. Here's a set of questions that cohere and are answerable. And at least for me, it doesn't always go from knowing what I want to do to writing a grant about it. Sometimes it goes from writing a grant to then saying, well, maybe that's what I should do since they've given me this money. So I think that's one of the challenges that I've had, at least.
Yeah. I'd say, in addition to what my colleagues have said, and I think alluded to, is finding a work-life balance, mentoring that for future scientists, being able to help teach all of these skills and then also how to survive as a graduate student. I talk now with my students, I'm not sure that I'd get in even now to the program that I got into. It just seems to increasingly, we talked about the threshold, the ratio of accessibility to funding increasing and growing broader, but the accessibility of being able to get into programs and even get started underneath this is getting far more difficult. So helping people who have really bent over backwards to get into our fields still find ways to not burn out so that they can keep asking interesting questions and kind of challenging the way things are done. And then I think just to speak to the particular funding for this project is finding agencies who are willing to take a leap with you if you're doing something really novel or translational that hasn't been done. You need data now to be able to do that. And so having access to seed funding as early career researchers to give a little bit of data to help support the new and innovative ideas we might have, you really need your foot in the door there to get going. So.
Hi, I was asking James Till about the fact that Canadians, he agrees that Canadians are not very good at publicizing what they're doing or anything. Basically, when Banting had to have a fist fight with his supervisor because they didn't think insulin was a very good thing to use in diabetes. What I wanted to know was, like, how are you supposed to know what's safe with foods that are safe to ingest and not cause diabetes? I gave up drinking 30 years ago, and I took up Diet Coke, and now I've got fatty liver from the high fructose corn syrup. You don't. I went to Tim Hortons for the Tim Prize, and they gave me a donut, which is poison for diabetes. I complained, and they hung up on me. How are you supposed to, why don't the diabetes research warn people about, what causes diabetes? Like, why don't they, before I get it, like they have a show on CBC about green tea causing the liver to get fatty with the green diet pills with green tea. That seems like these companies have multi-million-dollar advertising budgets to make the mass of people go in a group to take the stuff that's going to hurt them, and you don't have anybody from the diabetic organization coming out and saying, well, you shouldn't use these things, and don't follow the crowd, make your own decisions.
It's a nice linkage between our
I
thoughts.
was going to say,
Thank
there
you
you
for
go.
that.
I think one of the themes I hear you talking about now, and I've seen it a lot on social media in the last few weeks, given the circumstances occurring in the world that shall not be discussed, but it's becoming a good consumer of science, and that's a skill that we need to teach actively all the time, because you're right. We get all kinds of mixed messages and all kinds of data from all kinds of sources every day, both as scientists and also as, these are not my fields of expertise, so I'm a lay consumer of information in that area. And so to the extent that as teachers now and as scientists that we help people think about ways to critically be consumers of science, that's another really important hat that we wear that I don't know that I thought about as actively in my training, that now I really do think a lot more about at the undergraduate and graduate level of education. So I think it's a good point that you raised.
And I guess to talk to maybe promoting science, it's also part of the balancing act. Like if every study that was published made a grandiose claim, I think the actual important bits would be lost. Almost like if you walk down a street and there's too many signs yelling at you to do, walk, don't walk, you're not going to actually pay attention to them. So we need to have, I think, a nice skeptical view anytime something's published. And as scientists, we're almost innately trained to not scream too loudly about stuff, but let it kind of stand the test of time, just like insulin did, kind of impact people in a positive way.
All right, one last question here, yeah.
With FASD, if you catch it early enough, what is done with the children, and is it similar to the autism spectrum people?
Yeah, so at this point, from an intervention perspective, as of maybe the last time I reviewed the literature, there were only 36 good trials for interventions that showed really strong empirical grounds for what to do. We know that in terms of thinking about protective factors, the most important things are early identification, and not necessarily, I think, because that's important in and of itself. It's because early identification and a good assessment tends to identify early intervention needs. And once those are identified, it's more likely that a child will, and family, will, from a preventative side, perhaps be able to have more supports and change behaviors, but also to get kids into the types of early intervention programs that can improve outcomes. So I think for a long time in FASD, there was a scientific message. We were trying to tell people this is a real thing. There's real evidence. Because we didn't actually first diagnose FASD until 1968, 1973, when it was recognized in the medical literature. So for a long time we said, FASD is real, it's permanent. It's a permanent brain disability. And that communicated the message that, oh, there's nothing that can be done about it. So, now what? What we're actually now realizing and focusing more on is the shift in neuroplasticity. So some of the best intervention trials are looking at helping kids come up with adaptive and accommodative strategies that will allow them to better engage in the types of institutional opportunities we already have. So if we buff up a child's mathematical and executive functioning and attention skills, they may be able to benefit better from school. They stay in school longer. That's a protective factor against offending and antisocial peer influence. And so those are the areas that are really being focused on now. There's a whole array of prenatal types of interventions that are being studied really, hotly right now from a nutritional perspective, but not a great evidence basis around how to mitigate the effects of the actual prenatal alcohol exposure. So the money at this point is on early identification. And then the interventions are very similar to what you'd see in other neurodevelopmental disabilities. So dealing with symptoms and difficulties and having programs that will address those particular signs.
All right, well, thank you very much, and thank you. Let's join me in thanking our speakers. Thank you very much. Thank you, all token of our appreciation.
Thank you.
With your social skills and your very delicate issues.
Thank you.
And I hope you'll join us for a coffee. Thank you very much for here.